But the reason we're so interested in adenoviruses in terms of vaccines is that we learned early in the development of adenoviruses for gene therapy is that they're very effective adjuvants. And that's a challenge for us in gene therapy because the immune system sees the adenovirus, which interacts with antigen-presenting cells, such as dendritic cells, very quickly, and presents its antigens. That's a problem for the gene therapy field because the gene expression goes away, but it's very good for the vaccine field because it's a very, very effective adjuvant. And using adenoviruses, you can generate active immunity against the transgene, as the gene you put in the adenovirus, or the molecules, as I'll show you, conjugated to the outside of the adenovirus. The second platform, I'll show you a couple examples, is the adeno-associated virus. The adeno-associated virus is a very, very small virus, one of the smallest of the human viruses. There are 6 human serotypes and more than 25 non-human serotypes. And the way we use this virus is we get rid of all the viral genes, and we put in the genes that we're interested in. And the reason the adeno-associated virus is so useful is that unlike the adenovirus, which only expresses genes for 1 or 2 weeks, the adeno-associated virus, if you put it in the body in a place where the cells are not replicating, then you get long-term expression. In animals, we can get expression for the life of the animal. It's a very effective platform to deliver monoclonal antibodies, as I'll show you. Now, let me just make a couple of comments about the safety of gene therapy, because it's in the news every once in a while, and there have been some concerns about safety. As of today, there have been hundreds and hundreds of studies worldwide using these kind of vectors in thousands of humans, in everything from fatal disorders to normals. We did studies a few years ago using normal volunteers. There's been 1 death that was widely publicized with adenoviruses, and there's been 4 leukemias developed using retroviruses, which are viruses that insert their genome into the chromosomes. But at doses that we're talking about for these vaccine strategies, these viruses are very, very safe. So the 2 strategies I'm going to talk about are to deliver monoclonal antibodies and secondly to halt addiction. The concept in terms of delivery of monoclonal antibodies is we take the genes coding for the antibody, we put these genes into the gene transfer vector, and then the idea is to administer that to a patient. So one obvious question is, we have monoclonal antibodies, why bother? Why bother using gene transfer to deliver monoclonal antibodies? Well, one reason is that the typical half-life of monoclonal antibodies is only a few weeks. And so if you have a monoclonal antibody that you need to use over for a chronic disease, the concept would be that you can use gene transfer to strategize to provide persistent expression. But the second, and perhaps equally important, is that with monoclonal antibodies, of course, you have to give the antibody systemically. And you can't get the antibody into some places very easily. And so the concept of gene transfer is to provide high local concentrations, because that's where you would transfer the gene. So the strategy shown here on your left is using adenovirus to get rapid protection, that is for 1 to 2 weeks or months. And the other side is adeno-associated virus for long-term protection for years. And basically the concept is to take the genes coding for the heavy and light chains of the monoclonal antibody and put those into the coding regions of the virus. And the example I'm going to use is for those of you who know the cancer field and who know monoclonal antibodies, certainly know Avastin. Avastin is a product of Genentech and Roche, and it is a humanized IgG monoclonal antibody specific for vascular endothelial growth factor type A. Vascular endothelial growth factor is the factor that tumors use once they get above 1 or 2 millimeters. They have to have blood vessels to be able to maintain their size. So tumors are really smart, and what they do is they express vascular endothelial growth factor as the major mediator of making new blood vessels, and they attract in blood vessels from the local milieu. So the whole concept of Avastin, which is an antibody against vascular endothelial growth factor, is to suppress suppress the ability of tumors to grow blood vessels into them and therefore provide them with oxygen and nutrients. The Avastin binds to all of the vascular endothelial growth factor isoforms. There are several, and it prevents activation of the vascular endothelial growth factor receptors, which are called FLT1 and KDR. Avastin is the only FDA-approved anti-angiogenic agent for cancer therapy. Now, what are some of the concerns about Avastin? And the major ones are listed here. If you give it systemically, which is the way it's used, there's gastrointestinal perforation in 0.9 to 2.4%. 30% of when that happens are fatal. It interferes with wound healing because wound healing requires new blood vessels in about 0.8%. And pulmonary hemorrhage, 2.3% is higher in squamous cell cancers. The concept would be if we give these things locally, we could avoid these side effects. So the strategy that we use is we just basically took the Avastin sequence and we put that into an expression cassette, which is shown here. And one of the challenges is you have two genes. You have the heavy chain and the light chain, and you have to get those balanced. And that takes a little fiddling molecularly in terms of how do you do that. An example shown here, we're using one promoter and an internal ribosome entry site. And we put that in an adenovirus. I'm going to show you examples of both an adenovirus and adeno-associated virus. And then we have the virus coding for now Avastin. So the questions we're going to ask are, we usually think of antibodies made by B cells. Can we get other organs to make antibodies? Secondly, can we get high concentrations, local concentrations of antibodies? Can gene transfer be used to generate persistent levels of these antibodies? And finally, can we use it to treat cancer? So here's an example of the liver producing vascular endothelial growth factor. We don't usually think of the liver as making antibodies, but if we administered an adenovirus to a mouse intravenously, 95% of the vector goes to the liver. And if you look at the data on the right, which is serum anti-vascular endothelial growth factor antibodies in titer units, huge levels of this antibody are being made, as you can see, over a period of time with the blue dots going up over a period of time. What about the lung? We can take the same adenovirus, we put it down the lung of the animal intratracheally, and over a period of a few days, we look at the lining fluid in the lung, the epithelial lining fluid, for the production of the antibody. And as you can see, here again, very high titers with the yellow triangles going up over a period of time locally in the lung. What about the eye? And some of you know that the treatment now for age-related macular degeneration is another Genentech product, which is essentially, again, the Avastin molecule, but without the Fc portion on it. For this, you have to give injections directly into the eye once every month or once every couple of months. Would it be possible to get expression by just putting a gene transfer vector, in this case an adenovirus, and either put that intravitreally or subretinally? And as you can see on the right, we can get very high antibodies, particularly if we administer it subretinally. What happens is the virus infects the pigmented retinal epithelial cells, and that produces local high concentrations of the anti-VEGF antibody. Now, how long does this last? Well, here's an example if we give it intravenously, and it's unlike giving a monoclonal antibody. If you give a monoclonal antibody, you're giving it, and it eventually will go away over a period of a few weeks. But with a gene transfer vector, the vector essentially is a production system. It goes into the cell and is continually producing until the vector goes away. And here, even though the adenovirus goes away within a few weeks, here's out to 140 days, and we still have considerable levels in the serum of the anti-VEGF